Statistics is the discipline that concerns the collection, organization, analysis, interpretation, and presentation of data. And here we are looking at collection or sampling of your data. So sample should be representative of the population. Only when it fulfills this criteria that it allows you to extrapolate your conclusions, to generalize them to a larger population. Sampling could be further divided into random sampling, representative sampling, and convenience sampling. For random sampling, by its definition, it is done randomly. However, it is not haphazard, which means even when carrying out random sampling, you must ensure that every member of the population should have an equal chance of being selected. So in field stations where there is a physical gradient, and if your random sampling is not allowing you to get proper random samples, then you have to maybe use other forms of sampling. Next, representative sampling, where you purposely select cases to match the larger population. This is usually done for clinical studies or social sciences, where certain racial profile, gender profile is purposely selected to match the larger population. This is of course more difficult to plan, but the results will be easier to interpret. For convenience sampling, this is on the basis of ease of access, where if the samples do not differ from population and if the samples do not influence the outcome of the study, then convenience sampling works. Next, we look at the organization of your data. So usually to organize the data, we'll use a spreadsheet, for example at Microsoft Excel, LibreOffice, in order for us to arrange, rearrange, and tabulate the data so that it is easier for us to analyze them and also to graph them later. You have different types of data. For example, you have variable, constant data. Among the variables, you have quantitative, which is numerical data, or qualitative, which means either male, female, black, white. These are qualitative data. For numerical data, it also depends on the scale, the number scale that you are working with. If it's nominal scale, for example, labels and tags, for example, the number 10 jersey in any sports team, there is no weight and no numeric value in this number. Okay, so the numbers serve only as labels and tags. For ordinal scale, the numbers have weight, but there is no distance. So what I mean here is, for example, the placing, first, second, and third. So first is better than second, second is better than third. However, the distance between first and second second and third may not be equal. Okay, so that will be an ordinal scale. So for interval scale, there is weight and distance, but the 0 is arbitrary. So here we'll look at the Celsius scale as an example. So 20 degrees Celsius is 1 degree more than 19 degrees Celsius, whereas 21 degrees Celsius is 1 degree more than 20 degrees Celsius. So there is weight and distance among the temperatures. However, because the 0 is arbitrary, you cannot say that the 20 degrees Celsius is twice warmer than 10 degrees Celsius. Okay, so that represents an example of interval scale. In contrast, if you're looking at ratio scale, where there's weight and distance and has an absolute 0 point. So if A is 10 feet tall and B is 5 feet tall, then you can safely say that A is twice the height of B. So this is a characteristic of a scale that has an absolute 0 point. So this slide shows how you can group all the types of data that you have learned, and quantitative or qualitative. So if it's qualitative data, you can have ordinal and nominal scale. For quantitative, you can have discrete or continuous. For discrete, these are count data. So if for bacterial counts, it is actually under this category. For continuous, you will have interval and ratio scale. So this is how you can link all the different types of data together. Finally, when we have the data and when you have organized it, you need to analyze it. Okay. Statistical analysis can be divided into two main groups. One is descriptive statistics, the other is inferential statistics. So in descriptive statistics, which only applies to the sample studied, we describe the data that we have by calculating the mean, the median, the mode, standard deviation, standard error, confidence interval. These are numbers that will describe the data that you have. Next is inferential statistics, where we make conclusions and apply to the population. This is done by testing of null hypothesis, testing of no difference. That is, what is the probability of finding a difference this large in my samples by chance? That's why in t-test or ANOVA, if your p is less than 0.05, so that means the probability of finding this difference by chance is so small that you can reject the null hypothesis that there is no difference. Okay, and within inferential statistics, you can further divide it into association, comparative, and frequency statistics, where association statistics look for relationships, patterns, and trends, for example, using correlation and regression analysis. For comparative statistics, usually we look for similarity between groups and among groups, with t-tests for two groups, and analysis of variance or ANOVA for two groups or more. Then frequency statistics is usually for frequency and counts. We look for association or independence using the chi-square test. So thank you for watching, and please subscribe. Thank you very much.